Hello everyone, my name is Ahmed, and my paper is on Smoothened receptor mutations dictate resistance to vismodegib in basal cell keratosarcoma by Prykel et al. I chose this paper topic based on just the name. I was looking up stuff for the last Sonic the Hedgehog game called Forces. Remembered Andrew talked about Sonic Hedgehog in our last presentation, so I decided to connect them together and talk about an aspect of Sonic with some regards to medicine, and it seemed easy enough. It wasn't. I wish I chose something else, but it's too late now. This was a hard paper to study for and a rough week for me. Suffice it to say, this presentation is very rushed, so please go easy on me. So the main theme that ties my presentation together is to keep in mind, hey, there's this medication called vismodegib that stops faulty Sonics from causing cancer, but Sonic can still resist it. We're going to look at how Sonic works in detail, and how it doesn't to cause cancer, and how vismodegib can prevent that, and how Sonic can fight back, and what are some other ways of fighting against that. But before we begin, we have to go over some background info. So, Sonic Hedgehog is a protein that functions as a ligand for the hedgehog signaling pathway. Said pathway is responsible for all kinds of functions, mostly relating to the development of the embryos in the brain, nervous system, tooth formation, separations of digits. It is an extremely important protein and pathway for normal functions, as issues like mutations and misregulations of the pathway can be fatal, as one of the most common illnesses that can occur is cancer. The Sonic Hedgehog protein is named after the fictional video game character Sonic the Hedgehog, who is a blue anthropomorphic hedgehog with the ability to run at supersonic speeds. Now to quickly discuss the pathway. I'm going to plagiarize from Andrew. Thanks, Andrew. But I'm going to make my own diagrams in MS Paint. When there's no Sonic present, the pathway is considered inactivated, so the protein called patched inhibits smoothened, which results in Gli proteins getting cleaved into Gli repressor forms, which go on to inhibit transcription. When Sonic is present, it binds to patched, which doesn't inhibit smoothened, so Gli isn't cleaved and can go on to activate their respective genes and transcription. If something were to go wrong, like say smoothened doesn't get repressed due to a mutation in the pathway, then one of the potential outcomes is cancer. Speaking of which, smoothened is often the main target of treatments for cancers caused by faulty hedgehog signaling, because it's both pretty big and full of binding sites that normally intended for other proteins can be exploited by medication, and inhibiting it prevents many future agents from going rogue. One of the most prominent ways to inhibit smoothened is through a drug called vismodegib. Vismodegib is a cyclopamine, which is an alkaloid, a class of basic naturally occurring organic compounds that contain at least one nitrogen atom. It works by binding to one of smoothened's transmembrane domains and prevents the conformational changes to activate it. As of March 31st, 2020, vismodegib is still in testing to determine its efficacy. Now that all the setup is out of the way, let's get on to the experiment from 2015. So what is this paper all about? It talks about a specific set of tumors called basal cell keratosarcomas, BCCs, and some medulloblastomas. These are most commonly skin cancers, and they happen when the tumor repressor gene, patched1, stops working. Normally, patched1 represses smoothened, but an unrepressed smoothened can go on to cause the earlier tumors. This is known as a primary mutation. One way to treat this issue is with vismodegib, which inhibits smoothened, which can regress the tumors. However, another secondary mutation can occur, which blocks vismodegib binding and lets tumor progression continue regardless. The researchers want to look at how these smoothened mutations occur and to describe them. The first step was to obtain the tumors representing the two main kinds of mutations. To do this, the researchers obtained tumor samples from two patients. Patient 1 had confirmations of BCC metastasis in the liver, lung, and bones, and CT scans after 2 months showed disease progression at all sites, which was considered a primary mutation. Patient 2 also had BCC on suprascapular skin on top of the shoulder bone. Patient 2 was on 150 milligrams a day of vismodegib, which saw a reduction in the cancer for 5 months, but 11 months later, 2 subcutaneous nodules appeared in the area of the BCC and were removed and analyzed to suggest an acquired resistance. The researchers wanted to examine the mechanisms for the hedgehog pathway for these two patients. To do this, they performed patched1 sequencing. The pre-treatment of BCC liver metastasis of the case showing primary resistance but no patched1 mutation was observed. In contrast, patient 2's case, which, remember, was a pre-treatment primary biopsy of the case showing acquired resistance, showed the patched1 nonsense mutation, CAG into TAG, creating a premature stop codon, PQ84, in exon 2. This patched1 mutation predicts translation to a heavily truncated protein, causing loss of function of the protein and preventing inhibition of SMUDINT. The same patched1 mutation was also observed in the recurrent sample taken during vismodegib. Looking deeper, patient 1's case showed the SMUDINT missense mutation GGG into TGG at exon 9, which led to amino acid substitution G497W in the corresponding protein. Primary tumor and node metastasis also exhibited the same SMUDINT missense mutation GGG into TGG at exon 9. In contrast, patient 2's acquired resistance case didn't have such SMUDINT mutations detected. But the sample obtained from the recurrence on vismodegib showed the missense mutation GAC into TAC at exon 8, leading to the single amino acid substitution D473Y, confirming the acquired resistance to the inhibitor. This entire research section is summarized by this diagram the researchers made. A is our regular SMUDINT, C is our SMUDINT with the G49W mutation, B shows us the patched1 nonsense mutation CAG into TAG, creating a premature stop codon PQ84, and D shows us the missense mutation GAC into TAC at exon 8, which gets D473Y in. Now looking more at the mechanism, the researchers performed an in silico analysis, basically a computer simulation analysis, of the wild type and both clinically relevant SMUDINT mutant alleles. I'll skip most of the jargon and get to the interesting parts. They learned based on their simulation that the presence of the mutant residue results in the entire protein region undergoing a conformational rearrangement, which partially blocks the drug entry site. So this G497W mutation makes it harder for vismodegib to bind and work its magic. The researchers even called this, quote, a novel and effective mechanism of drug primary resistance. Looking at our other mutation, the researchers note how D473 affects things. Normally, there's a pivotal network of hydrogen bonds that keep the SMUDINT binding pocket in the proper shape and stabilizes vismodegib binding. But the 473Y residue has an aromatic side chain which affects said hydrogen bonding, making it harder for its inhibitor to work. So now that we understand the two mutations that limit vismodegib's effectiveness, what next? Well, this paper was written in 2015. A lot can happen in 5 years. So what are some new developments related to this? One option is to look at other treatments that inhibit SMUDINT instead of vismodegib. Kind of like how one antibiotic doesn't work anymore, you have others that can hopefully pick up the slack. This, of course, like antibiotic resistance, SMUDINT can adapt to all or more of these mutations down the line, which can further mess up inhibition. Not to mention that some medications are limited to particular cancers, so overlap is also somewhat limited, as well as side effects from mixing medications. But nonetheless, here are some alternatives that work in the same general way as vismodegib, but have some slight differences to hopefully overcome the resistance. This is from Targeting the Sonic Hedgehog Signaling Pathway, a review of SMUDINT and GLI inhibitors by Rimkus et al. From 2016. I'll mention some notable ones. Arismodegib is an orally available SMUDINT antagonist that has been shown to induce cell cycle arrest and apoptosis in a variety of cancer cell lines, currently in phase 2 testing. Saridegib is the result of chemical modifications to the base structure of cyclopamine. It has been shown to decrease tumor growth in lung and chondroseroma xenografts, but did not affect epithelial tumor cells, currently in phase 1 testing. Another option is to counter the counters, as it were. If you remember from this paper that I was assigned, one of the vismodegib mutations is D473H, and it has a potential answer. MRT92 is a drug that works by binding to multiple binding sites on SMUDINT. According to this 2016 paper, MRT92 inhibits hedgehog signaling by blocking overlapping binding sites in the transmembrane domain of the SMUDINT receptor. However, this paper doesn't say how exactly it blocks D473H beyond the whole binding thing. So how does it stabilize the hydrogen bonding? Maybe if I had more time to Google it, I would. But alas, we may never know. Another option is to bypass the middleman. This 2020 paper, the bromodomain inhibitor IBET-151 attenuates vismodegib-resistant esophageal adenocarcinoma growth through reduction of GLI signaling, shows that new players on the scene, like IBET-151 or bromodomain inhibitor, limits the growth for vismodegib-resistant cells for esophageal tumor growth. As you may recall from earlier, that vismodegib-resistant cells tend to have overactive GLI. So the treatment these guys found was you could treat vismodegib-resistant cells by going a few steps down the pathway and using IBET to inhibit GLI by inhibiting BRD4 as a workaround. BRD4 regulates GLI right on its promoters. What's significant about this approach is that most hedgehog pathway inhibitors work by directly trying to inhibit SMUDIN. IBET works level down by bypassing SMUDIN mutations. I don't have time to go into more depth because 1, I'm lazy, and 2, I chose a different paper as my primary topic to research. Now to come back around to our original paper to make some criticisms. But I can't. This paper seems done well. I couldn't find any major issues with their approaches, and I don't think I really can. Some potential nitpicks I am a little unsure of is the reliance on computer simulations to model and demonstrate much of their points instead of the more in vivo stuff, but they still do reference other researchers who have done the in vivo stuff, and I'm not really that familiar with how the computer simulations work, so I can't provide a critique on that. It doesn't really appear they use the control and brush past much of the mechanism with a heavy use of physics and kinetics as their explanations. But I completely admit that I could be wrong about all this, so take it with a grain of salt. So yeah, not much to complain about. Alright, let's wrap up this train wreck of a presentation. So, to conclude, I chose a really hard paper on vismodegib and how hedgehog signaling pathway can end up resisting it. Mutated SMUDIN caused by a G497W mutation causes a conformational change on the SMUDIN to prevent vismodegib from fitting in the first place. And a D473 mutation that makes it so binding sites are less stable, so it's harder for inhibitors to stick to it. This paper just looked at, yes, these can happen. Some ways to address this is by having more cyclopamines like esmodegib and ceridegib as backups, like how antibiotics work. Another option is to try and address the mutations. MRT92 can take on D473 mutations by binding to multiple binding sites. Or you can go a level down and use stuff like IBET to inhibit GLI. Thank you for watching this.